Hello everyone, I am going to be presenting about the plant growth promoting effects of rhizospheric root derived actinobacteria and Brassica juneca or brown mustard. The soil microbiota is a diverse and intricate community of microorganisms that reside in the soil surrounding plant roots. They interact with plants in numerous ways to enhance their growth and metabolic pathways. Among these microorganisms are plant growth promoting bacteria that produce auxins like indole 3-acetic acid and increase the concentration of glucosinolates in plants. IAA is a naturally occurring plant hormone that belongs to the class auxin, while glucosinolates are sulfur-containing glycosides derived from amino acids. Bacteria-produced IAA has been found to directly enhance plant growth and development by influencing physiological and biochemical pathways, whereas glucosinolates play a regulatory role in inflammation, stress response, metabolism, and antioxidant activity in plants. This ultimately increases nutrient availability and the supply of phytohormones that stimulate plant growth. It was hypothesized that the rhizospheric well-isolated endophytic bacteria will promote growth in Brassica juneca. The main objectives of this experiment were to isolate endobacteria from the roots of the host plants basil and mint, to confirm the production of IAA by these endobacteria, and to measure the amount of glucosinolates in the plant groups treated with the basil and mint derived endobacteria, the positive control people eczema, and the untreated group, which was treated with only soy broth. In the experiment, endobacteria from basil and mint roots were isolated using triptych soy agar plates, and they were purified by dilution streak plate method. The phenotypic identification of the isolated bacteria was performed by gram staining of the single most viable colony from each host, and the genotypic identification of those bacteria was carried out by biologs using inoculating fluids A and B. The mustard seeds were sown on sterile peat pellets for each of the 4 groups, and after 14 days of germination, each group was treated with its respective bacteria, and the untreated group was given only soy broth. After allowing the plants to grow for a week with the bacterial inoculation, they were harvested, and the height of each plant from each group was recorded, and one-way analysis of variance was used to analyze the data. And the combined matter of plants was also taken from each group. Salkowski's assay was used to assess the production of IAA by the basil and mint derived endobacteria, and each bacterial culture was supplemented with 6 micrograms per ml of L-tryptophan. Glucosinolates were extracted from the combined plant matter of each group by cold methanol extraction, and they were quantified using ultra-performance liquid chromatography in the presence of 10 millimolar cinegrin as the internal standard. In this experiment, people eczema was used as the positive control, and an untreated control consisted of only soy broth. The study found that the basil-derived endobacteria were gram-negative, whereas the mint-derived endobacteria were gram-positive. This Salkowski's assay used to test the production of IAA by these microorganisms revealed that the mint-derived endobacteria produced the highest amount of IAA in the presence of the 6 milligrams per ml L-tryptophan, and one-way analysis of variance or ANOVA of the plant heights post-harvest indicated that the mint-derived endobacteria significantly increased plant height that resulted in a combined plant biomass of 11.1 gram. It was the highest among all the groups tested. Moreover, ultra-performance liquid chromatography was used to quantify glucosinolate content in each group. Surprisingly, the untreated group had the highest glucosinolate content at 772.55 millimolars, exceeding the concentration of the standard itself. Meanwhile, the group treated with basil-derived endobacteria had the most types of glucosinolates compared to the other group. Overall, the results show that the actinobacteria derived from the mint root had growth promoting effects on the mustard plantlets, and it produced the highest amount of IAA in the presence of 6 milligrams per mil L-tryptophan. This study discovered that people eczema, which is a bacteria known to promote plant growth, did not have any beneficial effects on the growth of mustard plants in this experiment. However, the endobacteria isolated from mint demonstrated promising results during the IAA test and height analysis. Unexpectedly, the untreated group exhibited extremely high levels of glucosinolates, which could be due to natural stress or due to errors during the extraction process. These findings suggest that further research is necessary to explore the mechanism of plant growth promoters in different plant species, and it also highlights the significance of understanding the biodiversity of Endobacteria in agriculture and its potentials to enhance plant growth and yield.